afternoon, everyone, on this rather warm March 7th day here in Niagara. Welcome to our CCOVI Lecture Series speaker today. Our speaker today is Vincenzo DeLuca. He's a professor of biological sciences specialized in plant biochemistry here at Brock University, and also one of our CCOVI Fellows, and also has the distinct title of one of Canada's Tier 1 Canada Research Chairs that we have right here at our own Brock University. We're very, very lucky. In terms of Vincenzo's background educational achievements, he received his BSc in biology from Concordia University in Montreal, went on to get his master's in science and plant biochemistry from Queen's University in Kingston, and then went back to Concordia to get his PhD in plant biochemistry a couple years later. His research is focused mainly on functional genomics and metabolomics of secondary metabolism, not only in grapevines, but also in various medicinal plants, such as the Madagascar Periwinkle. He seeks to improve wine quality parameters and to enhance production of valuable medicinal products. In terms of our industry, the grape and wine industry, his research could be applied to look at helping to define most appropriate times to harvest grapes based on some of these secondary metabolites that Vincenzo discovers or researches, because many of these have impact on aroma and flavor within the grape that translate into the wine. Dr. DeLuca currently teaches topics in plant and microbial biotechnology here at Brock University, and today he's here to discuss some of the interesting biochemical changes that occur in Icewine grapes during that hang time on the vine until they're harvested once that temperature drops below -8 degrees Celsius.
Thanks a lot, Debbie. So it's a pleasure to be here today to talk to you about some projects that we've engaged in the last couple of years. And it's great to be doing it now because I have a cold that's starting to come on, and I suspect I'm going to be under the weather in the next couple of days. I decided to pick this particular title, Icewine grapes are biochemically active between September and harvest, simply because I thought it would attract a large audience. And of course, I'm not going to talk about this at all. No, I will. But I'd like to introduce the activities that are going on in my lab, at least as an advertisement for various things that we're doing. And today I'd like to introduce my research activities in the following order. The lab focus has been for many years now on a medicinal plant, which is the Madagascar Periwinkle. And this particular plant produces some very interesting anti-cancer compounds, the vinblastine and vincristine molecules. And these molecules are terribly complicated in their structures, and it's been a major challenge to try to understand how living systems are capable of making such exotic molecules in the plant. The work on Madagascar Periwinkle allowed us to develop a collaboration with a series of colleagues across Canada that gave us funding to do work that was funded by Genome Canada. And I'm just going to quickly go over that so that it gives you an idea of the number of different things that we're doing in the lab. I'd like to then focus on specifically projects that are on grapes. A few years ago, a company called Vinifera for Life, which was making grape powders from grape pomace, asked us to look at what are the natural products, what's the phytochemistry of some of the grape pomace powders that they were doing, and I'll introduce that briefly. We're also working together with Debbie and other colleagues in the Niagara Peninsula on the development of these products based on Appassimento grapes, and we're actually looking at some of the chemical changes that are taking place depending on the different drying techniques that are being used. I'm not going to talk much about that today. But it is a project that's quite exciting, and it's very nice for us to be involved in that project and to feel like we're contributing to it. We also have a project that was partially based on some work that we did in the Knit for Life project. And a company called Sweet and Sticky, who makes these Icewine syrups, has been interested in some of the phytochemistry of their syrups. And hence, we've been doing a project on the phytochemical analysis of Icewine grapes from late summer to winter harvest. And that's probably the area that relates to the topic of today's lecture. Finally, I'll go back to some interest we have on a family of proteins called acyl transferases that are really quite important in generation of various structural diversity in some of the organic molecules that they make, and are really very important in generating typical aromas, as you'll see when I talk about that. Okay? So we'll start with the medicinal plant story. There's colleagues at these various universities across Canada that are working with us on a large-scale metabolic experiment and a large-scale sequencing experiment. And the targets are 75 different medicinal plants. And these are some of the most important medicinal plants that have been used by human populations around the planet for the valuable molecules that they produce. And in our lab, specifically Dr. Sayaka Hatsumi Masada is involved in this project, and we have a couple of PhD candidates, Dylan Levac and Bonnie Salim, who are doing some pretty exciting work on the characterization of pathways for making valuable medicinal products. Specifically, the objectives of the project are to basically use genomics and metabolomics as a tool to more rapidly identify the biosynthetic pathways involved in making various valuable natural products. Here we can see various candidate genes, candidate metabolites that you can find that have various uses, either in medicinal applications or in producing things, for example, some of these bitter compounds, which many of you will recognize. I'm sure some of you are also beer drinkers, in addition to being wine drinkers. So elucidation of how plants manufacture those compounds involves understanding the genes and the biochemical pathways involved. Specifically, what's happening is the target molecules that are involved here are targets that have been identified by various labs across Canada. And the idea is to identify all the genes involved in manufacturing those particular metabolites. And then there are collaborators who are moving these genes into yeast. And the idea is to establish a proof of principle that microorganisms such as yeast could, in principle, be able to produce those types of metabolites. And you can imagine if you had a morphine-producing yeast, that would be quite interesting, for example. Specifically in our lab, our targets are this molecule called streptozidine that comes from the Madagascar Periwinkle. And if we're able to make streptozidine, there are 3,000 to 4,000 secondary metabolites that are derived from streptozidine that could theoretically be produced downstream of that. So that would be a valuable achievement if we accomplish that objective by the end of this project. And that's exactly what we're striving to do together with our collaborators at Concordia University. In principle, what happens is that researchers in the lab create libraries of candidate genes, and they do it through high-throughput sequencing. They harvest RNA from specific organs or tissues that are valuable in manufacturing the specific chemistry. And then this goes off to Genome Canada laboratories that do this massive sequencing. So new technologies called 454 sequencing or Illumina sequencing can give you, 454 will give you 1 million sequences in one round of sequencing. Illumina sequencing will give you 50 million sequences in one round of sequencing. So these are the technologies that in 5 or so years from now, they're going to be able to offer parents of young children, and they're going to ask whether you want your child to be sequenced or not. And 24 hours later, they'll have a fingerprint of the genetics of that child. And 10 years ago, nobody believed that, but in fact, that's exactly where we're going, and we're using this to generate these candidate gene libraries. And then we use these libraries to identify gene function. And we have a whole range of technologies that we've been developing in the lab that I don't really have a great deal of time to tell you about. But once those genes have been discovered, they are then sent off to colleagues that will integrate the genes into yeast. And then finally, by various combinations, they'll end up producing specific metabolites. So this is the great value of team efforts, where you have large skill sets in different groups that come together and create these synergies that allow you to do these amazing things that you can't do on your own. Now, apart from using yeast or bacteria as a production system, one of the things that our lab is particularly interested in is to take those genes and actually pop them back into plants and ask whether plants are a better manufacturing center for making these secondary metabolites. And in the end, the expense of production in this system versus this system will define which system will predominate from a point of view of where pharmaceutical companies are going to go in the manufacturing process. Okay? I'd like now to switch to some of the projects that we're doing in Grape and that have been funded by Vincenzo for Life. Kyung-hee Kim has been working on this project now for a couple of years. And as I said before, the data that was generated from this project is now generating new interest on the part of various companies in using our skill set to help them with some of the research that they're doing. And I just did a synopsis of what we found. And one of the interesting things that we did find is that when we look at grape powders over here, we were looking at specific phenols that are well known to the wine industry, at least: gallic acid, the condensed tannins, the catechins and epicatechins, the proanthocyanidins, and caffeic acid. And we wanted to ask whether the powder itself contains these metabolites when we compare the chemistry inside the wet pomace or inside the seed. And you can actually see that powders contain significant amounts of these phenols, which is what the Vincenzo for Life people were really interested in finding out what was in there. What was interesting is that metabolites that are extracted with methanol are restricted to certain levels, but then if you take the powder, the residue that's left over, and you hydrolyze that residue, you'll find that there's significant amounts of some of these phenols as well. And what we don't know is when people consume these powders, what happens to materials that are stuck to the cell walls. So there's a significant amounts of residual phenols, but we don't know how the digestive tract or the intestinal tract of the human being that consumes them affects them in that process. So we were able to show that indeed these types of phenols exist in considerable amounts inside the grape pomace in the powder itself. We went back and we also looked at anthocyanins because there's a lot of interest in anthocyanins like delphinidin, for example, because there's a lot of epidemiological studies that suggest that delphinidin is a powerful antioxidant. And there's some evidence that when we consume delphinidin, that some of the delphinidin actually goes into the bloodstream, and where it could have a biological effect before it's modified and secreted through the liver and through the kidneys and off to the urine. So you can see here that both wet pomace and pomace powder contain delphinidin, they contain cyanidin, and they contain malvidin. These are three different anthocyanins that you'll find in wine. And some of the residue is still in the pomace and in the powder. And it seems to come into the powder in an intact form. I think that was the whole idea of asking that particular question. What the particular value of this is another issue, right? Because how much do you need before you have a positive biological effect? That's an interesting question. One of the really interesting things that we found was that grape pomace powder contains large amounts of this triterpenoid called oleanolic acid. And we know that grapes synthesize these triterpenes very early on in grape development, right after flowering, as the grape starts to grow, and it starts to deposit large amounts of this triterpene on the surface of the skin. And it's interesting here that if you look at dry pomace, there's large amounts. We have quantitations of these if anybody's interested. There's large amounts of this in the dry pomace, and it's also found in the wet pomace. And you can see here it's in the grape seed, as well as in grape skin. Okay? So the powders that Vinifera for Life sells contains lots of this. What exactly is its biological role is another issue. Okay? But when Vinifera for Life asked us specifically, they said, I hope you find lots of resveratrol inside the powders. And most of you who make wine know that when you're making wine, you allow the anthocyanins to leach out of the skin because you want to make red wine. And in fact, a lot of the resveratrol is leached out as well. So we tried to find resveratrol. We found very little of it inside the powder. So you know, if you're selling a product like that, you can't make a claim that it contains this type of an ingredient if it's not there. Okay? We found very low levels of this dimeric form of resveratrol. And viniferin is very interesting in that the cosmetics industry is very interested in viniferin. You'll find that they can sell a bottle of this cosmetic for 80 or 90 euros. And you can imagine when you pick it up, it's a very light bottle. So there isn't much in there. But this viniferin is supposed to be good for the skin. So you can see that there's a range of possible uses of these compounds, but it's not in the powder of the grape pomace. If we now go to the part of the project that relates to the ice syrup, we also started to look at, one of the projects was to actually look at what happens to Icewine grapes between the month of August and harvest time, near the end of the year. And in this project, it was actually motivated by a number of things, and we were interested in asking questions relating to whether living tissues are involved in biosynthesis as they mature, and of course, they certainly are. Here's an example over here where you actually see different varieties of grapevines, and we know that once the grapevine undergoes the veraison period, they start to turn red, and they start to make anthocyanins. And I like this particular slide because it shows you that that the chemistry is varietal specific. Some grapes make no anthocyanins, like the Sangiovese, I think it's called. Sangiovese. Why do they call it Sauvignon? It's interesting. Sauvignon is, I think it's the Tokai of northern Italy. When I was reading about this, and actually this group is in Udine, where this work was done. But this slide shows you that there's differences in chemistry from one variety to another, and that the types of anthocyanins that are made are varietal specific. And they did a very nice diagram here to show you that the varietals that make red pigments are further up over here, and they tend to make this cyanidin type of anthocyanin. And you notice that the difference between the red and the blue is an addition of a hydroxyl group to convert it from a cyanidin to a delphinidin type of anthocyanin. And very nicely, the last three grapes over here tend to make lots of delphinidins, and you can actually see the increase in delphinidin production over there. And they showed definitely that the reasons why these grapes make lots of the delphinidins is because they've got a 3' 5' hydroxylase reactivity that is expressed and therefore is making this extra hydroxyl group, and you start to produce these particular pigments. So in this paper, they showed that there was a genotype-specific anthocyanin biosynthesis. They showed that the variation in the chemistry was based on differential expression of anthocyanin pathways, in other words, the terminal hydroxylase over here. And the color shift from red to blue occurred with increased hydroxylation of the anthocyanins. So here's an example of maturation taking place, and with maturation you activate biochemical programs for manufacturing anthocyanins. Now, the next question we had was, well, do living tissues actually make biosynthesis as they're dying? And I just decided to pick up this wonderful example that most of you who live in Eastern North America are familiar with. This is a sugar maple. I think our friends in Quebec are busy harvesting maple syrup, as well as in Ontario. And they're going off to the cabane à sucre, which is something I used to do when I lived in Montreal. And you can do that here as well, of course. And you see this shift of the sugar maple starting to turn red and yellow and orange. And what's interesting is, as the season gets shorter and the sugar maple starts to undergo senescence, one of the strange things that happens is it starts to manufacture anthocyanins. And it's counterintuitive. Why on earth would a living system be making new products when it's about to die? And people who are interested in photosynthesis, one of the hypotheses that they raised was that anthocyanins were being made as a way to protect the photosystems that are involved in photosynthesis. Because the anthocyanins act as an absorber of light and UV light, and therefore, because the light intensity is still quite high in the fall and it's colder, photosynthesis drops. And because photosynthesis drops, it's unable to cope with the high light intensity unless you make anthocyanins. And that's very interesting. But as you can see over here, you also have another very interesting scenario which shows you a leaf cross-section. And in fact, anthocyanins are simply being made in the palisade mesophyll as opposed to any other cell types within the leaf. So it's very highly programmed, and only certain leaves, cells within the leaf are able to make that. But here's an example of biosynthesis being activated during the death program. I like this other example, this quite dastardly plant known as tobacco. And you know that we used to grow lots of tobacco in southern Ontario. And when the tobacco is ready, what we do is we pick it and we stick it into rooms where they are allowed to dry. Okay? And as they dry, they go from a nice green color to this leathery brown color, which is then used to make cigarettes and cigars and pipe tobacco and so on. And it's interesting that tobacco has, certain tobacco cultivars are able to convert the molecule nicotine to the molecule known as nornicotine. And what's really remarkable about nicotine is, those of you who probably know this, but nicotine is actually made in roots. And once the synthesis of nicotine has occurred, the nicotine is transported all the way up to the leaves and deposited there. Okay, so the biosynthesis of nicotine involves more than one organ, and it involves long distance transport. And in fact, when during the summer, what farmers do, they will decapitate the flowering parts of the plant because that triggers a wound response, and that increases the synthesis and transport of nicotine to the leaves. which is why they want to have higher nicotine levels for producing cigarettes. Now, there are certain tobacco cultivars that are known as converters. And interestingly, the converters, as they undergo the transition from green to yellow, in other words, they're dying, they activate a cytochrome P450 dependent reaction that will remove the methyl group from nicotine to produce nornicotine. Why on earth would you activate that biosynthetic process is a mystery, but it's true that nornicotine is a powerful precursor of nitrosamines. And you can imagine that the tobacco industry is not too keen in having tobacco lines that produce a lot of nornicotine. So of course they're very interested in this particular reaction. They want to prevent it from happening so that these nitrosamine precursors are not produced within the tobacco. So here's another example of a tissue that's dying and is now triggering another biosynthetic step. And these types of activities triggered our own interest in asking the same kinds of questions about what was happening in Icewine grapes. And I know that for a number of years now, there was an interest in whether Icewine grapes that are left on the vine are actually different from an Icewine grape that you harvest in September and stick in the freezer and make Icewine from. And we had some resources to do a preliminary experiment a year and a half or so ago. And Kim was doing this as a sideline. And we were very surprised to see, and you can see we don't have a complete time course over here. We chose Vidal and Cabernet Franc as the grapes that were being used to test. And I'm sorry, I can't tell you much about what the metabolite is because there may be some commercial value in this. But what you notice is that in both Vidal and Cabernet Franc, we see the level of metabolite A rising continually from month to month up to 20 micrograms per gram dry weight in the case of Vidal, and up to 60 or so micrograms per gram dry weight in the case of Cabernet Franc. And we just chose one metabolite, malvodin, over here, to actually show you that in the case of malvodin, it stays pretty constant. So there's specificity in the observation in that there's this new metabolite that starts to be produced and seems to be produced specifically during the period of Icewine grape production. Okay? And when we got this result, we started talking a little bit with Debbie Ingalls about this, and so we started to get involved more with this organization, which is Sweetness City and the Icewine syrups that they were producing. So metabolite A increases about fourfold by December in Vidal grapes. Metabolite A increases about threefold by the end of November in Cabernet Franc grapes. We notice that the other metabolites have not changed substantially during this period. And our conclusion was that these mature grapes are biochemically active. Because you have to find a way, how do you make that metabolite? You have to have biosynthesis. And so here's another example of a metabolite being produced in spite of the fact that the tissue is either dying or is dead. Of course, grapes are kind of interesting in that perspective because they're full of carbon, right? They're loaded with sugar. So you've got the carbon resources for biosynthesis. All you need is somehow the environmental conditions are triggering this biosynthetic pathway. And we thought, well, metabolite A is an important physiological marker for real versus fake Icewine. And hence why we think that this might be a good way to market maybe what real Icewine is. So we repeated this experiment. You do it once. Unfortunately, with grapes, you can only do it once every year because you got to wait for the growing season. And here we repeated Cabernet Franc. And what you notice here is that once again we see an increase in the level of this metabolite the increase is quite a bit higher than the previous year. And we noticed that the metabolite increased to about 94 micrograms per gram dry weight by December in these Cabernet Franc grapes. The conclusions we can make here is that the reproducibility of the results from 2010 confirms that metabolite A can be used as a physiological marker for real versus fake Icewine grapes. And then we did the same thing with Riesling grapes. And what we notice is that mature grapes of different varieties may all be biochemically active to make this metabolite in spite of the fact that they are dying. And there appear to be variety-specific differences in the amount of metabolite A that is being made during the production of Icewine grapes. The reasons we say this is that you notice here we're up to about 300 micrograms of this metabolite in Riesling grapes. In Vidal grapes we were at about 20, and in Cabernet Sauvignon grapes we were somewhere between 60 and 90 micrograms. So the reproducibility of these results, once again, in this third variety, suggests that metabolite A can be used as a physiological marker for fake versus real Icewine. What we're doing right now is actually monitoring gene expression. We know the pathway that's involved, and so we're actually using transcript analysis at each of these stages of development and actually seeing if the transcript is going to be found, which would represent the biochemical pathway that's involved. And finally, we're going to actually do enzyme assays to show that the genes that are expressed actually produce a protein product that's responsible for the biosynthesis. Okay? Finally, I'd like to tell you a little bit about some work that Brent Wiens, who just finished his master's degree in our lab, and we decided to tackle these acyl transferases, but this time in vinifera varieties, as opposed to the work we had done earlier in Vitis labrusca. And you can see that our interests in Vitis labrusca were based on the foxy flavor of some of the labrusca materials that's responsible for making these wonderful breakfast products, but not very good in making wine. And when we look at Vitis labrusca, the reasons for this is that they make this product called methylanthranilate. And many years ago, our group discovered that the production of methylanthranilate was from this coenzyme A ester called anthranil-CoA and methanol. And this is another really interesting example of dying tissues, because the methanol is actually coming from the breakdowns of the pectin that's inside the cell walls of these plants. And yet, then it's used in the synthesis of this methylanthranilate, which accumulates within the grapes. And this particular enzyme is a member of what we call the bad family of plant acyl transferases. This is a former colleague in my lab who decided to coin the name, and it's stuck now since the early 90s. And what you notice is that with this particular acyl transferase, it starts to be expressed 10 weeks after flowering. So it really coincides with verisome, and we can actually see the transcript level rising, we see the protein levels rising, and we actually see the enzyme levels rising. Okay? And of course, the other thing that I'm not showing here is that methylanthranilate starts accumulating, and as a result of that. So the process is coordinately regulated and it's verisome specific. And if acylated molecules of methylanthranilate don't convince you, if you look at strawberries, they make ethyl butyrate. Apples make butyl acetate, isoamyl acetate. This is, banana aroma. And so all of these metabolites are made via specific acyl transferases. So acyl transferases are really important in imparting particular aroma profiles to the fruits that are characteristic for those aroma profiles. Now, this is a really busy slide, but it's kind of a wonderful slide. Brent took the 88 different biochemically characterized bad acyl transferases and organized them into this kind of phylogenetic tree from 1, 2, 3, 4, and 5. 5 over here is a unique member. There's only one, but obviously there may be a lot more hidden away somewhere in nature. And you notice that many of these genes in this tree over here are involved in acylation of anthocyanins. For example, they malonylate this sugar position on the 3 glucosylated anthocyanins. We know that the substitutions with these molecules tend to stabilize the anthocyanins. So there's some real value in knowing a little bit about acylation of those positions. If you look at the second family over here, we have alkaloids. We have this morphinan that is acylated, this indole alkaloid that's acylated, or this acetylated benzoic acid. So benzyl acetate has quite an aroma associated with it. In this third class, you've got various phenols that are being acylated that are part of this particular family. And this last one is a very interesting one. And you can see the methylanthanylate enzyme is here. And this is the Taxol molecule. Those of you who don't know Taxol, it's a very important anti-cancer agent. And you'll notice that there's several acylations. There's 1, 2 acetylations. There's a benzoic acid being added over here. There's an amino acid called phenylalanine over here. And another benzoic acid moiety. Those are 5 different acyl transferases that are responsible for making Taxol. So this is benzobenzoate, another floral compound. I hope that this phylogenetic tree shows you how important acylation is for generating this type of interesting biodiversity that plant systems are able to generate. Biological activity and sensory activities. So we said, well, what does grapevine look like? Because the grape genome has been sequenced, we went back and looked at what were the bad acyl transferases that were found in wine grape. And then the question was that we were interested in, do these reactions have biological roles that may be important in determining sensory qualities? This is not an easy thing to do, but we got started on this. And the question then, of course, is will there be any practical uses in grape cultivation or breeding programs? So Brent, once again, just took the grape acyl transferases and showed that there's members of each clade. You can see here clade 1, clade 2, clade 3, clade 4, and clade 5. So there's members of each clade in grape, and of course, there's not one of them whose biological function we know. So none of them are known. There's a lot of them there. There's 51 of them. And so what we decided to do was to go to this clade because the Vitis labrusca methylanthanylate enzyme is over there. And we decided to focus on this gene over here, which is about 95% identical at the amino acid level. So you'd say, well, if there's any gene in grapevine, in Vitis vinifera, that will do this reaction, it's going to be that one because it's so similar. Well, the cloning of that gene, we obtained this gene from Sauvignon Blanc variety. And you notice that the Sauvignon Blanc has all these various acyl alcohols, which are basically various chain lengths, various aliphatics. And they are an important component of what Sauvignon Blanc is in terms of the flavor molecules that are produced within this system. So because of the fact that these molecules accumulate, we thought, well, does this enzyme catalyze that particular reaction, that acylation reaction? So the first thing we did was we went back into the grapes and we harvested protein. These are known as ammonium sulfate precipitations. and we give them substrates. We give them this cis-3-hexenoyl alcohol and acetyl coenzyme A. And then we run GC-MS, and you'll notice that in the first ammonium sulfate cut, there's no reaction product being produced. But in the 30 to 70% ammonium sulfate cut, we get this nice little peak over here, which is coincident with where cis-3-hexanyl acetate is going to migrate on the GC. And it's got the same mass. When we use buffer, which is a negative control, in other words, no protein, we see that this peak is not produced. So it was telling us that there is probably an enzyme activity within Sauvignon Blanc that is responsible for this so-called CHAT activity. Okay? So Brent went back and did the same experiment now with the clone gene, expressed the protein in bacteria, and here's the experiment where he adds the cis-3-hexanol and acetyl-CoA, and you have a reaction product. You notice how much cleaner the results are over here. If he removes the cis-3-hexanol, so you're removing one of the substrates, you lose the ability to make the product. If you boil the protein, so to kill the enzyme, well, you're not going to make any product. And of course, this is another control that researchers like us like to do, which is the vector that was used to engineer the sequence and the protein. And without the sequence inside the vector, we make no reaction product. And here's the standard. So this is very clear that indeed the recombinant enzyme from Sauvignon Blanc is responsible for CHAT activity. When we tested anthraenoyl-CoA or benzoyl-CoA, which works with Venus labrusca, it's not active. So a few differences in amino acids in the two proteins give us a difference in biochemistry, okay, and the characteristic chemistry that you find in Sauvignon Blanc versus Venus labrusca, okay? So the conclusions here is that recombinant Sauvignon Blanc clone catalyzed the formation of cis-3-hexanyl acetate. And compared with Venus labrusca, the Sauvignon Blanc gene displays a completely different expression profile. I have not shown you that. And then the results suggest that the Sauvignon Blanc protein has a different biological role in Venus vinifera compared to the Venus labrusca gene. Now, I just want to go back for a brief second to anthocyanin biosynthesis, this wonderful pathway that produces red versus blue versus grapes without any color. And you notice that the difference between these two reflects, is based on the hydroxylation pattern, and oftentimes you will get glucosylation at this position over here. And a couple of years ago, there was a paper that was published in agricultural food chemistry, and I talked a little bit about this a couple of years ago when I gave a talk to you. And it related to the inability of European grapevines to produce the 3,5-diglucoside, and the fact that this measuring the levels of this has been used to classify them according to their varietal origin. And of course, if the 5-glucoside was found, one was suspicious that the grape was contaminated with North American germplasm. Well, this particular study actually showed that the Venus vinifera has a 5-glucosyl transferase inside its genome. The problem with it is it's got 2 mutations, and therefore doesn't produce an active protein. And they showed that if they corrected these 2 mutations, in bacteria, of course, they were able to get a revertant. And the fact that you have 2 mutations suggests that it's very hard for the system to recuperate that function. And hence, Venus vinifera does not have 5-GT activity. And one of the questions I had in my own mind was, is this something that these mutations, did they occur in wild ancestors, or were they a consequence of domestication of grapevine? And that's still a question that remains to be answered. Now, the reasons why I showed this was because when they identified where this mutation, this mutant gene is on chromosome 9 of Vitis vinifera, it was remarkable that the Sauvignon Blanc chat is right next to the Vitis vinifera 5GT. And therefore, the selection as for 3,5-diglycosides as a diagnostic was not accidental, because if you would get 3,5-diglycosides, then it was likely that you also inherited AMAT, which comes from the foxy aroma coming from North American germplasm. And I thought this was quite interesting, that Sauvignon Blanc chat would be right next to it. And now that we know that Sauvignon Blanc doesn't make AMAT types of activity, it makes the aliphatic alcohol types of activity. Okay? And so I explained how the co-localization of this gene would explain the genetic linkage between the two traits in hybrid cultivars, and hence why we use it as a diagnostic. So I want to summarize now the things that we've done over the last couple of years. When we look at grape pomace powders, we were able to show that they contain lots of gallic, proanthocyanidins like the catechins. They contain some anthocyanins like delphinidin. But, as I said before, they have lots of this oleanolic acid. It's interesting, it would be interesting to see if this has any biological relevance that might make grape pomace powders valuable sources of this particular molecule. For the phytochemical analysis of Icewine grapes from late summer to winter harvest, we've identified a metabolite that we call metabolite A that can be used as a physiological marker for real versus fake Icewine grapes. For the acyl transferase story, we've now identified a function for this Sauvignon Blanc clone that makes cis-3-hexanyl or other aliphatic acetates, which we're now testing. So, and to differentiate the vinifera versus Zabrusca situation. I'd like to acknowledge the financial assistance of Vinifera for Life and the Ontario Centres of Excellence that permitted the grape pomace work to be done, and also Sweet and Sticky that helped us get Federal Development Art Grant together with Debbie Eagles. It's a collaboration we have between our two labs, and she's doing something different in that project. And finally, I really want to acknowledge the people who did the work. Kyung-hee Kim, who was really indispensable for a number of the projects that I described here, and Brent Weens, who did the acylation, and of course, he was rewarded with his master's degree recently. And finally, I think that's it. Yes, thank you very much.
So we've got time for some questions from the group, if any come forward.
There's one online from Fabian Gagne. He wants to know if you have a publication or report coming on the phytochemical analysis of Icewine grapes, and if so, when approximately?
Right now, we're looking at perhaps putting together some intellectual property, and I'm hoping that's going to happen in the next couple of months. The publication, if the intellectual property comes out without any problem, should come soon after that. So I would say, hopefully, in 2012.
Just the second part is, who are the owners of this intellectual property of metabolite A?
Well, at present, the owners of the property, anything that's done at Brock University belongs to the university. And usually what happens is we negotiate licenses with interested parties. Since the project was done with Sweet and Sticky, they would have the first right to that intellectual property before we would open it up to other people who might be interested.
James Hill from Philly States Winery. Icewine grapes continue to develop in a sense past November 26th and past December 10th. progress further past those dates that you have there? Is that what you conclude?
Yeah, I think there's going to be a time when you start to lose metabolite A. And so if you wait too long, the levels are going to start to drop, either due to the grape undergoing complete degradation. And this is very interesting because if you look at Riesling, for example, it's a very delicate grape, and therefore its ability to withstand is going to be shorter than other grape cultivars. And so you have to be, from varietal to varietal, the timing of that is going to be different, as you will probably acknowledge. And so we do see drops later on in metabolite A.
Will you continue further showing what those changes are? For example, there's certain years that we don't get the cold weather that we want.
Yeah.
And we know that there's individuals that may harvest much earlier. And obviously the characteristics in the wine are just not as intense. When I harvest, we usually do our Riesling grapes first. We do our red vinifera second, and our Vidal we've harvested as late as March.
Right.
So I guess what I would be interested in finding out is the flavor components. Would you be able to tell me that the is metabolic A? If I picked Vidal in later February and we make it into an Icewine, we ship it to China, and all of a sudden we're confronted with a knockoff, and to prove that it's a knockoff, we're looking at a means of trying to prove that. Now, whether it does this any good or not in China, who knows?
Yeah. I think it could be used as a marketing tool, a very effective marketing tool, because it's a real difference that we can document in the levels of this metabolite. If you saw, there were grapes that were harvested in the month of August and September. Well, those were frozen, and then they were analyzed several months later. You stop making metabolite A if you freeze it. So if you leave it on the vine, the levels of that metabolite are changing continuously. And I would put it to you that it might be a good way to find out whether you can use metabolite A as a diagnostic for the best time to harvest your material in terms of the different types of wine that you want to make. And that's something that needs to be tested, where you would actually do, when the level is at 20 versus 40 versus 60, what are the differences in the types of wine that you're going to be able to produce. And as far as I'm aware, this is one of I'm not aware of other examples of specific metabolites that seem to follow the changes from month to month with the grape changes that are going to take place with Icewine production. And if you notice, the results we got last year are different from the results we got this year, which tells us that it's a good, it might be a good marker for environmental, the stage of the environment that we're sampling that year.
Exactly.
And so that's why I wonder if it would be valuable to monitor that metabolite from year to year to establish trends and patterns that would then allow you to translate that into the quality that you generate from one year to the next.
And prime example would be this past year, 2011, we've seen the dehydration happen quite quickly, but because we didn't have cold weather, we weren't getting that color change from green to brown, and hence we were waiting for that period of time for it to occur, and as a result, some of the vinifera reds were completely desiccated to the point that there was almost nothing left.
We're good for making raisin bran.
Exactly.
Yeah, it's a very interesting environmental barometer, it seems to me. When Kim was observing the results that she was observing, she was saying, well, we're getting the trend, but the levels are different. And last winter was very different from this winter. And it's not surprising then if something is sensing the environment in terms of, and it also does suggest that there's biosynthesis taking place, because it seems to be different from one year to the next, and whatever triggers that biosynthetic pathway is affecting the process.
Not to monopolize your time, just one last question.
Yeah.
Can metabolic A be artificially added?
Well, I think it could be artificially added. The question is whether you can artificially add it to the grape itself, because the place to do the analysis is in the grape. And it wouldn't be that easy to do.
I guess from my perspective, I'd be actually looking at the finished product, the wine. So
Right.
is metabolic A then actually in the finished wine?
The finished wine, the levels are going to be very low in the finished wine. So the place to do the analysis is in the grape that's been harvested for making Icewine grapes. So the idea would be the producer would ship the grape that's being processed for analysis, and you get a stamp saying the metabolite A is where we expected it, and therefore this is an authentic product.
Okay.
That would be the way to do it.
Do we have any other questions from the group?
Just one more. Kareem wants to know, is metabolic A important from a sensory?
Is metabolite A important from a sensory standpoint? I know that there are some sensory attributes associated to it. They would be probably minor, but they do have some. They do have some effect on the color of the wine, for example.
Well, if you'll please join me in thanking Vincenzo DeLuca for an exhilarating talk today. So many different facets of your program. But also, I hope you can appreciate from this work that as many answers as you've received or determined so far, there's lots more questions to be looked at and applications of the approaches that you take to research. So we just have a little thank you gift on behalf of Kevin to share. Thanks, and for those of you locally, just a quick announcement. We actually have another lecture tomorrow afternoon from a specialist, a yeast specialist from L'Allemande, Italia, in the northern part of Italy, that will be coming here to Covey to talk about a few different topics. Brettanomyces yeast being her area of specialty, but also some non-Saccharomyces yeast that they're now exploring uses of in the production of Amarone style wines. So there may be some interest from the wider community here to come and listen to Tiziana talk tomorrow. It's at 11:15 right here in this room. Thank you.